It all started a few years back when we were discussing at length with Karsten about the role of tubulin PTMs in regulating key molecular processes, and we were wondering if this could play an important role in spindle assembly. Then we decided we could not address this only between the two groups, but we needed to team up and take a much broader approach. So we got in touch with many different groups, and we managed to arrange a very broad team of expertise ranging from synthetic biology to modeling, but also high-end microscopy, 3D reconstruction, and also different model systems, which was providing an extremely powerful platform for the training of new PhD students.
There are two types of cell division, meiosis and mitosis, but in both cases a spindle is formed, which is a molecular machine that separates DNA into two new cells. And my job was to figure out if the spindle is different in these two different cell division types, why is it different and in which way.
During one of the phases of mitosis, which is called anaphase, the spindle goes from a length of 3 micrometers to a length of approximately 12 micrometers. So the length increases 4 times, but the total polymer remains more or less constant, which means that microtubules are lost as the spindle elongates. My project was to try to understand how this process is coordinated in the cell. So in biology, we build models to explain our observations. And with these models, we can make predictions that we can verify experimentally to falsify our models. Sometimes the process is simple and we can either get a positive or negative result from the experiment, but sometimes the complexity of the system requires the use of either a set of equations or even a computer simulation to address the problem.
So I was working on the interaction between dynein and the kinetochore, and I was trying to answer the critical question of how we transition from metaphase and to anaphase during cell division.
The question I was trying to answer was, what is the profile of post-translational modifications of tubulin in the meiotic spindle?
My research is focused on a group of proteins related to microtubule cytoskeleton called as MAPs, or microtubule associated proteins. And in my project, I developed a purification-free pipeline to study around 50 of these proteins, and most of them are associated to many cellular processes such as cell division and are also linked to many human diseases.
So my PhD project was to study temperature control applied to cell division. So my main task was to develop microfluidic tools for the biologists to study cell division in the microscope.
My project in the divide network was to fully reconstruct the mitotic spindle in mammalian cell. And with this big data, we are trying to understand what is the ultrastructure of the mitotic mammalian spindle, but also focus on the connection between the structural connection between the kinetochore and the spindle core.
For me, I think it was a great opportunity to have collaborations with other members. So I built this image analysis software for Anna, which is one of the students in the network, and I also worked in doing a simulation for a project in the lab of Thomas Surrey, which is one of the PIs in the network. So for me, I greatly benefited from this possibility of collaboration.
Well, the fact that there were so many of us, so not only were there 10 additional PhDs besides me, but also their supervisors, and this allowed to always have a fresh and new perspective on my project, and I think that was really helpful.
Well, I think that obviously it gave us moral support, and difficult times were easier when you get to share them with people who live the same things as you do. But also it gave us technical advice because we were not necessarily working on exactly the same projects, but some of the techniques were similar and we were able to exchange on that, so that was very helpful.
So sometimes biologists have some difficulties in order to go farther or beyond in their research. So here engineers help them to unlock those technological issues that they have.
In the network, there are not only PhDs and supervisors from the academia, but also from the industry. And this has given me first-hand information and knowledge about their experiences and their career paths. And on top of that, Divide has organized some courses where we were introduced to people from a wide variety of backgrounds who are occupying professional niches which were before unfamiliar to me. So that was very informative.
The Divide Network provided us training, but more importantly, I think it was also training on things that gave us a different approach, like for instance working on modeling or communication, things that I would not have necessarily gone to in the first place.
So actually for us to organize a conference was quite of a challenge because none of us has done it before, so we had to learn everything from scratch. Besides, I think one additional complication is that we were 11 people in different institutions that had to coordinate in order to solve the small issues through the way. So this was a bit, like a tug of war, but at the end it was super rewarding because we could see that the people enjoyed the conference and the attendees did tell us that it had been a great experience for them. Besides, we also got new contacts, so we also got to know new researchers, and all in all, you felt like you did a great job.
The outcome of the ITN is, of course, science. We did a lot of scientific research, and now many papers will come out of it, in addition to all the PhD theses of the students. But in addition, I think it provided extremely good training platform for the students to train them on many other aspects, not only science, but soft skills.